So the title of the talk today is Relating Drug Molecular Data to Indications using Canonical Component Analysis and Linear Models. So here today I will present our effort in method development to quantify the molecular mechanisms and to associate molecular mechanisms into drug indications. So for drugs, we make observations of molecular mechanisms in two terms. One is the known functional target. The other is the off-target or unintended bindings to all other human proteins. So for the unintended binding, we define the terms promiscuity. And for the known functional targets, we analyze its effectiveness or the drugability. So for this, we expect to have a large or complete data set where the experimental assays are available for an ideal way is we have a complete matrix between the drugs and all the human proteins. However, such things are not available. So we developed a computational method to calculate the two terms based on limited resource of the experimental data, the promiscuity and the drugability. From there, we associated the two molecular mechanism terms to drug indications. Here we analyze the side effect and the dose. So as I just said, that the experimental data are not really available. So we had hope on Campbell, on binding DB. We were thinking, okay, there are a large amount of assays there. So when we really look into it, we found that most assays are focused on the known drug and the target interactions. And there are a lot of data of low confidence and repeated on the known interactions instead of testing on the novel ones. So we cleaned and filtered the data. We found that on average, there are only 15 unique assays for each drug and 2 assays for each proteins. And most of these interactions are the known drug targets interactions. For example, this has been recorded in the drug bank or stage. So then we searched for literature. We found another 2 broad scale in vitro pharmacology profilings. One is by Novartis. They analyzed about 200 drugs against 73 targets. The other effort is by Pfizer. They analyzed 92 targets. Again, most of the targets here are known drug targets inside of the novel off targets or the proteins people don't know they actually interact with the drugs. So then we go to the computational method as an alternative. Computational method can be not perfect, but in our research, we have found that if we start from a solid experimental data as a starting point, we actually can generate biological insight with pretty good precision. So this one. So as I just in the first slide, I showed that I need to talk about the 2 algorithms where we define the terms of the molecular mechanism. One of it is the promiscuity. This is the method we have developed in 2011. So that was a method to compare 2 drug binding sites to measure the similarities of binding site. With that, then if we know that 2 binding sites are similar, then we assume that they share a drug or a small molecule ligand. So we have built a data set about more than 2,000 binding sites that found in a non-redundant site of human proteins. So given the drug binding site, for example, celecoxib in COX-2, we look at the binding site, we compare the binding site against the 2,000 human proteins. Then we look for the similar microenvironment, which here represented as the colorful spheres. And then we identify the similar environment, then we add up them together so that we are able to rank the 2,000 human proteins to calculate a potential of binding to celecoxib. So conceptually, we're able to generate a profile of each drug against the 2,000 human proteins. So in this figure, I show you the promiscuity signature or the off-target profiles of each drug against the 2,000 human proteins. So here the 5 rows are found inside naproxen, celecoxib, and so on. So all these 5 enzymes bind to COX-2 as their functional targets. Using the traditional structure-based method, it's very hard to detect the subtle difference between all the file binding sites as shown in the right panel over there. But if we compare the binding site against the 2,000 human proteins, they actually generate a different promiscuity signature. And this signature actually also suggests that the drug classification, for example, the naproxen is a rapid inhibitor, which is the first row, and then celecoxib is a irreversible, and the other 3 are reversible inhibitors. So with that, that is, given the drug, we build the profiles. We have the vectors of its binding potential against all the human proteins. So we validate this promiscuity in 2 aspects. First, we look for the experimental assays again. So we calculated interactions between drugs and proteins, again, between 200 drugs and 2,000 human proteins, which is a very large number of interactions. Among them, we're able to retrieve about 0.5% of the interactions where the experimental data is available. So then we plot those IC50s found in the Campbell against the calculation against our predicted affinity. We see that the predicted affinity can suggest the potential binding. In the second aspect, we first decide a heuristic cutoff where we're able to count the off-targets, so calculate the promiscuity index using a certain cutoff. There, using that cutoff, we claim that 15% of drug bind to 20% of the human protein. That's the estimation of the overall 200 drugs against all the human proteins. So according to the Novartis experimental report, they say 20% of drug bind to the 10 out to 20% or 70 profiled targets, which using their 5 macromolar IC50 cutoff. So we have based on these two validations, using the very limited resource of the experimental data, we kind of use these 2 as a confidence builder. See, right now we're able to generate profiles of all the known drugs, small molecule drugs, of their promiscuity profiles. So then we move on to the next step. We want to relate this promiscuity to the indications. This is a very simple experiment. We simply count the number of the side effects recorded in the CIDR, and then we plot against the promiscuity index. We found this rough correlation. That this is not a surprise, but we want to build a method where we are able to detect a higher resolution association between that side effect and the promiscuity. So the method we use here is the canonical component analysis, or the CCA. We use that to associate the molecular data we calculated using the computational method and the drug indications. So the CCA is a method where it can associate 2 multi-dimensional variables. For example, we have the molecular data X. Here we use the essential protein bound to the predicted bound to the drugs as X, and we use the indication side effect as Y. So the algorithm is projecting the X and Y into a certain subspace. In each subspace or the component, the method is searching for the basis vectors alpha and beta so that the projected matrix of X and Y look alike most, or the projected matrix show the correlation between the projected matrix are maximized. So in that, after we search for the basis vector alpha and beta, in each component, we're able to do 2 things. First, we're able to predict Y given the new X. Second, we're able to look at each component and using a cutoff to decide the significantly association between specific attributes in X and Y. So that I showed in the first application where we want to predict Y using the X. So for the Y here, we use a side effect. We first built a subset of side effect based on CIDR, which is the most popular resource for side effect. We removed the side effects that are most frequently observed. We only want to predict side effects that are not easy to predict. So for example, the fever and the headache, we removed those first. And then for molecular data, we could use the predicted promiscuity or here I use essential protein bond. That's the same thing here, which is the off-target bond. And we can also extract the known target information from stitch and drug bank, so that we compare the performance using different molecular data to predict the wide side effect indication. So then the comparison, the performance showed that the promiscuity, which is the computational predicted the molecular data, actually drives the prediction. So for many drugs, the FDA drug label doesn't have a clear label of side effect. For example, some of the NSAID is one of the drugs of interest in our lab. So it's a very simple example. So starting from that drug binding side, we predict the yellow box, which is the off-target, their potential binding sites there. The red ones we are able to have the validation. And then from there, we can derive the green box, where is the indication. In the second application of that CCA is that we look into each channel or each subspace, and where we could decide a cutoff of each attribute. For example, like certain weight for the attributes, the alpha and the beta assigned to each of the essential proteins and the side effect. We remove the first channel specifically here because in the process of deriving these basis vectors, alpha and beta, we use the SVD so that we could extract the dominant feature of the data, which represents the data frequency. We remove that because we want to look into the biologically relevant inside of the data frequency. So here, from there, we are able to associate a certain side effect and the genes, the off-target we predicted. Then it's very, very hard to find a validation of such association without the drug information because the association is simply between the genes and the side effect. So we have to map that back to the drug to find the validation. Here is an example. For this side effect, the master irregularities, we predicted 7 genes, and we found 7 drugs associated in this subnetwork. And then in this figure, I show the validation as a confidence builder that 6 of the bindings between the targets and between the predicted genes and the drugs have been found with IC50 better than 10 macromolars. And then for the 7 genes, Novartis have an experiment where they identified 10 genes for this specific side effect of the menstrual irregularities. They confirmed the 10 genes actually have the causal relationship to this side effect. In their 10 genes, we actually are able to predict 4 of those, where is the orange colored ones over there. So this is the example where some of the relationship can be validated with the limited experimental resource. But then for a lot of them are still new. So we at the end, we actually extract more than 2,000 triplet sets of drug essential genes and side effect. Only about like 2% of them we're able to find the validation. But then we analyzed this association. We found that they are actually biologically meaningful. For example, we look at the essential genes or proteins enriched in these associations where they are significantly associated with the side effect. We found that they enrich the nuclear hormone receptors and the transcription factors. These genes are on the upstream of the pathways, which may cause the cascade reaction of the downstream. So instead of the enzyme and kinases, these genes are likely to cause the side effect in our analysis. So far, we have talked about the algorithm, the method for determine the molecular mechanism, the promiscuity, and how we relate the promiscuity to side effect. However, we didn't talk about the other important terms of drug, which is the functional target. So the functional target of protein, that is how effectiveness the target, the protein actually take the drugs in our body. So we previously developed a method, drugability. We found that this method can be used to evaluate how optimal a drug adapts its functional targets. We use that to represent the functional target's effectiveness. And then, so for indications, because we analyze side effects of NSAID, we found that dose is actually a very important indication for drugs too. So here we analyzed the MRTD, which estimates the upper limits beyond which a drug's efficacy is not increased, and the side effects begin to overweight the beneficial effects. So in analyzing the NSAID, we have found that, for example, celecoxib is a low-dose drug compared with other NSAID. And then when we do the calculation of the promiscuity, celecoxib is a drug of high promiscuity. And its functional target, we analyzed its binding affinity between the functional target COX-2 and the celecoxib. We found that this is a very effective functional target for celecoxib. That is, celecoxib has the high drugability and high promiscuity. So that explains the fact that celecoxib is a low-dose drug because it's able to be low-dose because it's high effectiveness, and it has to be low-dose because it's high promiscuity. So then we want to build a linear model of these two terms to determine the dose. So we have a collaborator at University of New Mexico, Tudor. He collected the dose data for us, about 200 drugs. So we plug in the dose data into the promiscuity and the drugability, where we're able to find the significant linear relationship. And then we also found that combining two terms is better than one. Using this linear model, we're able to reevaluate the dose of drugs where the severe side effects are found, and then provide the guidance for drug discovery. This is the last slide here. Here's a short example, the prediction using the linear model to predict the drug dose of 16 NSAID. So using the linear model, the predicted dose at it's
the best thing is you can comment. I think people saw that there was a correlation, so maybe you can.
Okay, so over there is there. So where we're able to predict the dose for 16 NSAID, and the best performance one is the red cluster over there, where we actually found that the dose prediction using the linear model based on the two molecular mechanism terms, promiscuity and drugability, where it works best at these two situations. One is that its starting point is a good experimental structure where the COX-2 co-crystallized NSAID structure are available. We start there and then calculate the molecular mechanism terms. The second observation is that if the drug is a selective inhibitor where the binding site of the experimental, the binding site binding affinity matters a lot, where then the dose prediction are accurate. So that this makes sense because a selective inhibitor, its binding pattern, binding affinity actually is a key step of this of drug's action. So that's the last slide, where is the application of how the dose prediction works. So overall, we have built this computational framework where we quantify the molecular mechanisms, promiscuity of drugs, and the druggability of the drug targets, where then we use these two computational terms and then relate it to the phenotype, the drug indications. And then for the last slide, I really want to show the pictures of people I want to acknowledge. I want to thank Ross. Ross is not here this year. And also the people who works in our lab, also Tudor from New Mexico. Thank you.
Thank you. Yeah, we'll take one or two questions.
Nice talk. There were, on the US market, 3 COX-2 inhibitors, valdacoxib, celecoxib, and rofecoxib. So hoping to see in that slide, maybe you can bring it back. Do your predictions differentiate between the 2 that were withdrawn from the US market and celecoxib?
Celecoxib is on the market. And then the second one, oh, I can't use this very well. So one is, most of them are withdrawn, and one is on the European market, but not on the, what's your question?
Does your model differentiate between celecoxib and the 2 that were withdrawn from the US market?
So this one is on the market, and I think this is also on the market, but this one is on the European market, and this is withdrawn. I'm not very sure. Yeah.
I guess the question is, where's Vioxx?
What?
Where is Vioxx? That's rofecoxib, which is a very famous COX-2 inhibitor.
I don't have the structure. If I don't have structure, I can't do things here.
Another question?
Yeah, hi, nice talk, man. So your talk was, side effects predicting for a given drug. What are the different proteins or the binding site that it binds to and the side effects? Mm-hmm. So if you look at the, other side of it, could you comment on, based on similarities existing at the binding sites, if you look at the other side, you could also comment about the drug repurposing for the same drug.
Yeah.
So were you able to find out some, different binding sites from completely different proteins which you could use it?
Yeah, the repurposing is also an ongoing project in our lab. Here? Okay, so your question is this computational method able to be applied to a repurposing project, right? Yeah. Okay, yes, we do have the project where we apply the method where we're able to compare the binding site. And then, so for example, if you have the target genes, you want to find the drug, then you compare that genes, search for the potential site, and then scan against all the known drug binding site. Yeah. Thank you.
Question.
Thanks for the presentation. Just a quick question. Do you integrate 3D models, and if yes, what are your criteria to detect a binding site on other human proteome?
How we detected the binding site?
Not how you detect, but you are using 3D structures, yes, to detect binding site of targets.
Yes.
My question is, do you integrate 3D models of protein structures to improve the coverage, the structural coverage of the human proteome?
We didn't. So for the binding site detections, we actually have a very simple rules because it's one protein. There is a statistics analysis said 85% of the drug binding sites are actually located on the largest pocket in the protein. We just use that simple rules. We search for the largest pocket. Is that your question? I
I
couldn't hear very
think
clearly.
The question was whether you used predicted structures instead of...
We don't.
yeah.
Yeah, because also one of the take-home message is we want to start from a very solid experimental data from there because we don't want to introduce multiple layers of uncertainty in computational method. Thank you.
And perhaps, sorry, just one more precise question. Do you take into account missing atoms for this detection or not at all?
The missing items?
Yeah, in 3D structures.
I did. Yeah, I did for some of that. But, I think I did for all of those because we actually have a toolbox where we process all the structures.
Okay, perhaps we can talk about it later.
All right, thanks again.
Thank you.